Right, today we're going to continue with aerobic cell respiration, and we last left off on a video that ended with glycolysis. And so, if you recall, glycolysis ended with two molecules of pyruvate, pyruvate being a 3-carbon molecule. So at this point, we're going to continue to oxidize pyruvate by taking out electrons occasionally. So here's pyruvate, and there's two pictures of this because both of them from glycolysis are taking place. So you can kind of imagine that these things are happening simultaneously. I'll probably focus only on one at a time. So pyruvate at this point is going to transition to what's known as an acetyl group. But in that process, there are things that happen. The first thing that happens is it is going to be oxidized. It's going to lose an electron. And where does it lose it to? The electron carrier of NAD+. So NAD+ is going to go in. It's going to remove a high energy electron. And in that process, it takes other things like a hydrogen, and it turns into NADH. So we've made NADH before. That happened in glycolysis, so here we have another one. But as well, we also notice over here, we're going to lose a carbon dioxide. That's why we went from a 3-carbon molecule to a 2-carbon molecule. And that carbon dioxide is released. So if you look at the formula for cell respiration, C6H12O6, which is our glucose that started glycolysis, we're going through aerobic respiration, so we're going to need oxygen. We haven't needed it yet, but we will release CO2 because that's where it comes out. It's the first time it comes out is what's known as, it doesn't have a scientific name. You can either call it this conversion step, or I also call it the prep stage. We're basically prepping this molecule to enter the next major process in aerobic cell respiration. And eventually, we are also going to make water. So at this point, we have a 2-carbon molecule called the acetyl group. And the acetyl group is going to enter into the second major process of respiration, which is going to be located down here. We'll get to it in a minute. But this acetyl group can't enter on its own. It's going to need the help of an enzyme. So the enzyme actually is going to join up, and this picture shows that it joins up here, and it's called coenzyme A. So together, the coenzyme A and the acetyl group join a molecule called acetyl-CoA. This molecule is capable of entering into the second phase of aerobic cell respiration. So the same thing happened over here. Again, I'm calling this the prep stage because we basically prepped pyruvate to turn into acetyl-CoA, and this occurs within the matrix of the mitochondria. So there had to be some movement of this pyruvate because this was made in glycolysis, and glycolysis occurs in the cytoplasm. So this pyruvate had to move from the cytoplasm into the matrix of the mitochondria. So here's a little scorecard. Things that went into here, we started with the pyruvate, and we had 2 of them. We ended with the acetyl-CoA. We had 2 of them produced. We also had some NAD+, 2 of them, one on the left, one on the right. And what they get turned into is the reduced form, which is NADH. So this has that high energy electron. We made 2 of them. And as well, we have this byproduct right there, CO2. And we produced 2 of them as well. We did not make any ATP in this phase. But we are ready to go into the next section of aerobic respiration. This is known as the Krebs cycle. So the Krebs cycle is taking the end result of the prep stage, which is acetyl-CoA, and it's going to enter into an already turning Krebs cycle, but it has to enter at this exact location. There's a molecule that we somewhat regenerated. Think of this like the dark reaction. The dark reaction regenerated RUBP. This molecule is called oxaloacetate. I know the spelling is a little bit hard to see there. Oxaloacetate. Oxaloacetate is going to join with the acetyl-CoA, and once that happens, the role of the enzyme is over. So this coenzyme A can actually go right back up and help another acetyl group join. This acetyl-CoA now comes down here, so it's kind of just this one big cycle. All right? So once the acetyl group that now had the help of the enzyme joins with oxaloacetate, it forms a 6-carbon molecule. You can see the 4 that are there, the 2 that are there, forms citric acid. Sometimes this cycle is actually called the citric acid cycle as well. There are kind of two alternative names for it. And this goes through a series of changes. This is a very detailed picture. You can kind of see at each step what happens, but we're really focused on what are the molecules that are coming out. So we notice that nothing changes in terms of molecules between citric acid and this intermediate 6-carbon molecule. But after that, we have a formation of NADH, which is important, and we have a release of CO2. This molecule then does a somewhat identical process. It releases an NADH and another CO2. So we went from 6 carbons to 5 carbons to 4 carbons because each one was lost within a carbon dioxide, while producing more energy or high energy electron carriers. This 4-carbon molecule then forms a direct molecule of ATP, which is really the basis for this whole process, is to make ATP. So that's good that we made one right there. But it continues on, and in that process, here we have another formation of a new molecule. It's abbreviated FADH2. It has the same role as NADH. It's an electron carrier. So the oxidized version, which I've always kind of said is more like an empty vehicle, and this molecule, the FADH2, is the vehicle that has the passenger in it, where the passenger is a high energy electron. So the FAD goes in, and it removes an electron from this reactant, and it forms FADH2. The product of here continues on. Nothing really happens in this stage that we need to worry about. The next stage, we have another formation of an NADH. And at that point, we are back to oxaloacetate. So this whole cycle regenerated to what we started with. But in the process, we made some very important molecules, such as NADH, ATP, and FADH2. But we also made some byproduct CO2 molecules, which we know are byproducts that get released out of a mitochondria. So this is happening simultaneously because, once again, one glucose produces 2 pyruvates, which then go into 2 acetyl-CoAs, so 2 Krebs cycles happen per glucose. And that's kind of how we're tabulating everything, is based on a glucose. So let's look at reactants. So we had the acetyl-CoA. That really started this process, and we had 2 of them, one on the left, one on the right. Byproducts, things that came out of this process that are part of equations. The only thing is this is CO2. We had 1, 2 on the left, and then we'll have another 2 on the right. So this is 4, which makes sense because the acetyl group has 2 carbons, 2 carbons. So that's 4 carbons total, and now we had a byproduct of 4 carbons that came out. Now let's get into the energy or electron carriers. So we had NAD plus as a reactant. We actually have 6 of them. 1, 2, and 3. Then there's double that because it happened again over here. They each formed into an NADH molecule, so you would have 6 of those. And then you also had ATP, but the reactant that you formed ATP from was ADP. That was 1 on the left, 1 on the right, 2 total. So that then made us ATP, 2 total. And we also had FAD. So that was the oxidized version. That was what went in. And then what comes out is FADH2. And that was 1 on the left, 1 on the right. So that's also 2 here, 2 here. So for a total ATP, we have made 2. So at this point, we're pretty much done with making everything. We have to now go into the final phase of aerobic respiration, which is known as the electron transport chain, which seems recognizable as the ETC from photosynthesis. So the idea here is that all the molecules that have been carrying those electrons are actually going to come to the inner membrane of the mitochondria. So if you remember the mitochondria as this bean-shaped structure, there's the outer membrane. The inner membrane has all these folds called cristae. And what we've done is we've enlarged this portion of the membrane, of the inside membrane, to the level that you can actually see the phospholipids. So the perspective is that above this membrane, this section up here is called the matrix, and that's been identified right here. The space between the inner membrane and outer membrane, I've exaggerated that. On a true mitochondria, it wouldn't have that much space, but for the diagram to be helpful, this is called the intermembrane space because it's the space between the membranes. So what we have here is an electron transport chain. And embedded in the electron transport chain, I'm going to kind of highlight them to make them a little easier to see. There are 3 hydrogen pumps. So I'll say H pump 1, H pump 2, H pump 3. If you recall from photosynthesis, pumps act in an active transport mechanism. So that means that they're going to go from low to high concentration with the use of energy. Just like the photosynthesis hydrogen pump, it utilizes energy from high energy electrons dropped off an electron transport chain. So NADH, which has that high energy electron, is going to drop the electron off. So I'll kind of exaggerate. There's the electron. Imagine that. The oxidized version, where it lost the electron, now is NAD+. That NAD+ can now go back to the Krebs cycle. Pick up more electrons. So it gets recycled over and over again. Going back to this electron, this high energy electron is now going to bypass the first hydrogen pump. Just like the hydrogen pump in photosynthesis, this hydrogen pump says, I'm going to utilize some of your energy and I'm going to pump a hydrogen from low to high. So look at the direction I've moved, from matrix to the intermembrane space. So that then assumes that this is the low concentration side, this is the high concentration side. So in terms of hydrogen ions, there's a difference in concentration gradient. There's not as many over here. So I'll kind of make it like that. The electron still has some energy. It continues. It bypasses a second hydrogen pump. This pump says, I'm going to do the same thing. So a second hydrogen ion gets pumped into the intermembrane space. It continues on. A third pump utilizes it. So there was enough energy in this one electron to help pump 3 hydrogen ions. And these hydrogen ions were up here. All right? They didn't have to come from this NADH. Sorry. This NADH doesn't supply all the hydrogens that are being pumped across. Once this electron is removed, this NAD+ is removed, and possibly that hydrogen ion that just came from here is also now just kind of floating around in the matrix. Eventually, it might make its way to a pump and make its way over to the intermembrane space. But don't get confused. A lot of students think that that hydrogen, which is no longer visible, must be the hydrogen that went through. It doesn't have to be that way. So this one electron pumped 3 hydrogens across. These hydrogens don't want to be on this high concentrated side. So look at this picture over here, where the hydrogens that are in high concentration, there's my 3 that I just pumped across. What they're going to do is they're going to migrate to an ion channel. This is ATP synthase. Again, the same enzyme we looked at in photosynthesis. This ion channel is going to allow the hydrogen ion to diffuse passively, but also activate this enzyme. So this enzyme is going to churn and make ADP plus a phosphate into ATP. So the analogy has been that NADH is like foreign currency. This one NADH that we made over here, this was responsible for pumping 3 hydrogen ions across, which potentially 3 hydrogen ions then can go to ATP synthase and make 3 ATP. So 1 NADH is really equivalent to 3 ATP, which is the currency of our cell's energy. NADH is more foreign. It's not the form of energy we use. We use ATP. And finally, FADH2 is the other electron carrier. But notice where it comes in. It comes in after the first. So its electron only bypasses the second and third pump, therefore only allowing 2 hydrogens across. Thus, the currency is only that 2 were really here because of that 1 electron. So 1 FADH2 is really only a currency of 2 ATP. So I'm going to stop there because I'm getting to the 15-minute mark, but we'll pick up the end of this process and anaerobic in our next and last video.